So this is my third seminar here from my postdoc here at the department. And it's going to be the first seminar that's not about Brazil. So finally, I can show you a bit of what I've been doing here for the past year or so. And the name of my project, the biggest aim, or the broad project name is Plant Communities Through Time. So I'll make a small project report for you, mainly now about this one paper that was published, that is Weather Fluctuations Drive Short-Term Dynamics and Long-Term Stability in Plant Communities, a 25-year study in Central European dry grassland. Okay, so this seminar, as I told you before, I will tell you a bit about the broader project, that is Plant Communities Through Time, some goals about the project, and then we go for the main part of what I'm presenting today, about the paper with introduction, methods, results and discussion, some conclusions. Also something else about this paper that I have to show you today. And then a bit of the next step of what I'm going to do next. So my biggest project here that I have since I started is called Plant Communities Through Time. And by the name you must guess that is about plant community dynamics, and one way to evaluate that, and that's the way we are using now, is by means of repeated vegetation surveys in permanent plots. So for this broad aim, we separated in two different or specific aims for two different time scales that we are evaluating these dynamics. The first one is a large scale. I can hear my echo, people. Does anybody have your mics on? Okay. And the second one is in a thinner scale, where we check within-year dynamics, also relating to phenology of the plants. Where we resurvey the same plots monthly for a year. So here is just a small scheme showing the importance or why what we can learn if we resurvey the same plots for different times. So here you can see the dots represents these permanent plots every time you survey them. So in the dots you can see that the communities are varying a lot, and also you can check that sometimes they are more close or they are more different from each other. So if you sample only once and then again, like in this kind of resampling technique, you will get only two separate pictures of your community. You won't have the whole story of the dynamics of this community. And this is why having different frequencies, we can see what's actually happening throughout many years, or if we change years here for months. So then we go directly to the main aspect of this seminar, that is the paper in which I present the results of the first aim, which is for the surveys that are done annually for 25 years. So I would like to start this presentation by talking about this very important issue, that is climate change. And it's known by most of you or all of you that is one of the biggest threats to biodiversity today. And we have the increase of extreme climatic events and increasing, these fluctuations are increasing both seasonally and between single years and consecutive years. I can hear some noise. So it is necessary to describe and to understand how these fine-scale changes in vegetation are following those oscillatory weather patterns. And especially for temperate grasslands, which are very dynamic ecosystems. The community composition and richness, they change a lot, both in space. You see small patches not very far from each other, having totally different species composition, but they also vary a lot through time. And this variation through time, they depend a lot on the fluctuations of the weather conditions. These variations are also combined with intrinsic population and community dynamics, not only related to weather. And these variation in environmental conditions are very important. They are key for this type of environment because of mechanisms that maintain biodiversity, supporting the co-occurrence of species. This climate fluctuation prevents the competitive exclusion between species that co-occur in a site. So they allow the species with different strategies and from different climatic domains to coexist. So if a species is more related to a dry weather, it will increase its dominance in that given year. But if in the following year the weather is not so good for it anymore, then it will decrease and be slowly substituted by other types of species. And if we have this random or fluctuating weather where one year is drier than another and vice versa, then you can keep this non-equilibrium coexistence between the species. And this mechanism is specifically true for dry grasslands in these shallow rocky soils because they also shift a lot, but they are long-term stable. Several studies already shown in this kind of permanent plots, the same as we did, that because there are plants with different different life strategies, they respond different to this climatic variation, and so they can maintain this high diversity. So the goals of our study is to use two datasets. The main one is the dataset of species composition and abundance from a dry grassland in permanent plots for 25 years. And we also use non-permanent plot data from the same area that were sampled several times since 1930. And we want to answer the following questions. If in the long term there are directional changes in dry grasslands plant community, what is the extent of these interannual fluctuations in species composition and relative abundance? And whether these year-to-year dynamics in the community depends on weather conditions of the current and the previous year. And if so, the species with these different life stories and different responses to climate will show different patterns to these weather fluctuations. So we studied, our study area is in the Evin Hill, that is part of the Pavlov Hills in southeast of the Czech Republic. The climate in the region is subcontinental. The annual temperature varies from 8 to 9 degrees. We have January is the coldest month. It can have the average -1 to -2. In July, the temperature can get more mild. The precipitation, the annual precipitation is about 500 millimeters, with being around 300 in the growing season. But one thing I want to highlight is about this temporal variation in precipitation between seasons. And also that long periods of drought are very common. And the altitude of the Avon Hill is 549 meters. Here I have the data, the climatic data for the whole period of the 25 years. A little bit more than that. And you can see how it is changing, like how it varies throughout the year. And also how we have years that, for example, are way more wet than others. And years in which we have some drought periods here, represented by these red parts in the graph. The dry season or the droughts are most importantly during the growing season, of course. And here the average to show how the average should be and to compare to the other years how they don't follow this average most of the times. One other thing that I want to highlight here is that we had to pay a lot for this data and now they are available for free. But okay, this is the temperature for the same region. And you can see that in the past 28 years, the temperature is increasing. Those are averages per season also. Okay, and the vegetation in the area is situated in the Pannonian Phytogeographic Province, and it's part of this Euro-Siberian forest steppe region. As I told you before, there is high species richness and high diversity between small areas, and also because it mixes some species from sub-Mediterranean with continental steppe species. And the specific part where we had our plots were in these south-facing slopes of the alliances Festucan valenciensis and Bromopanalsi Festucium platensis. So about our data sets now. The first we are going to call the historical data set. That is this data set that we have since 1930 by many different researchers that we could get from the database. And one thing to highlight here is that they were not done in the specifically same plots. So this is not a repeated vegetation survey. This is just historical surveys in the same area, but not in the same plots. Now we have the permanent plots that was set by Irka or Kiri. This was set in 1993, and it was 7 quadrats of 4 by 4 square meters in this dry grassland alliance that I told before. And you can see by the picture that it's very rocky, so there are rock outcrops covering more than 10% of the area. And the samples were done by all vascular plants. They were visually estimated, the cover. And it was done every spring in May or June from 1993 until 2018. For those species, we separated them in three categories based on the lifespan. Annuals, short-lived, that combine both biennial and short-lived monocarpic perennials, and the ones that we defined that was perennials, that would get, let's say, more than 2 years of weather affecting that individual, while these ones would, even if they are perennial, they are not so long-lived. Here there are some examples of each one of these life strategies. And also what we did with those climatic variables that we bought, we calculated average per season for 2 years before. For example, if I want to see what's affecting the sampling of 2005, sampling here represented, I will get data for spring 2005, and the winter was defined by December 2004, and the other months for 2005. I will get the whole year of 2004 and 2003. So this is the whole data set of weather variables that I want to compare with one sampling. So of course they will overlap, because there was another sampling here on May, but then this data will get also 2 years below here. I don't know if it's very clear now, but then with the figures, I think you will understand it more clearly. The analysis that we did for the historical data set, we just run a PCOA ordination. We also run the same PCOA ordination for the permanent plots, in which we have all the plots 25 times. And we also calculated variation partitioning based on this partial distance-based redundancy analysis. And we try to decompose the variability in community composition based on plot identity that will give us how much this variation is explained by the space, also by time, defined by the year of the evaluation, and what was stochastic. As a second step, we try to decompose this time, the explanation of time in the variability. We decomposed into directional and non-directional parts. How we did it? We simply entered year as a variable 2 times. Year was entered in the model 2 times. Once as a quantitative variable, and second as a categorical variable. So we had these 2 predictors, which was year. But here we tested as if year was not related, like one year to another is not related, and as categorical is as they are not related, and quantitative is that the time is countable. Then, separately, we also run another variation partitioning of this temporal variability. What is explained by temperature and precipitation 8 seasons before, these 2 years of weather conditions, what is explaining this variability in time. Also, if this is not very clear now with the figures, you'll see that it's going to be clear. So now the results. First, we have here the historical data set. So the colors indicate the older ones are purple and the more recent ones are yellowish. So there is kind of, I don't know, separation for at least these more modern ones. But what I wanted to show you here is that we also plotted the permanent plots. So we just put the the initial one and the final one, just to see where we are in this long-term database. And apparently there is a separation between them, but here we have to remember that those plots were defined by what this or that researcher was looking for, so maybe we don't know which bias or which were the aims of them, so we cannot say for sure how these changes can be related, if it's a change in the community or it's just a change in the placing of the plots. Here are the ordinations just with the permanent plots. Each colorful line represents one of the plots. With the star is the beginning and the arrow is the ending point. So we can see that there is it's very clear separation between them. We can see that each plot is very separated in axis 1. And the axis 2, you can also say that you see some trend, but we don't want to interpret this as so clearly, because most of the time there is a very random pattern, and just in the last 3 or 4 years they are actually separating from this randomness. But we will discuss this further. Here, just how many species we found, how many were in each of the life forms, and you can see the complete data share here. Okay, so this is the result for that variation partitioning, which was kind of confusing, but now I hope I can explain you better. So first we wanted to see how much of the variation of the communities were because of space, which is how plots are defining the variation, and the space have a big importance, as we just saw in the PCOA. This is mostly that axis 1 represented here. And here is how time is defining the variation. And time is explaining a lot, like more than space actually. But when we separate how much time is important in non-directional and the directional way, non-directional mean year as qualitative variable, and directional means year as a quantitative variable. So only this small part is telling us that there is a trend varying from the year 0 to the year 25, while all this other explanation is saying that you have communities varying together year by year, but this variation is not leading towards something different. This year is in one state, and maybe in 5 years it can be very similar state. It doesn't mean that every year is changing a bit more towards something. I hope I was clear. Then we can discuss it further. So, and the third part of the variation partitioning, which is including weather into the model, we here just separated how much weather was explaining the variation in the current year, that is the year right before the sampling, and the previous year. So both of the years explained a lot, actually. But the current year explained a little bit more. And of course, there is a part that's not explained by the weather. So here is the plot of this RDA. And it's very interesting to see, because we can see here how these weather variables are influencing the species in the plots. So here we plotted only the species. And here the weather variables. The blue is for precipitation and red is for temperature. And the darker shade is for the current year, and the more lighter color is for the previous year. That means this 2 years ago, before the survey. And what we can see here is a separation between these groups or life forms. We can see that perennials are mainly linked to these negative values on axis 1, while the other types, annual and these short-lived, are more related to positive in axis 1. So we can see that wet and colder summers are more related to perennial species, while wet spring and warm winter and also wet winter are related to this species, while hot and dry summer is related to this share of the species. Okay, and this is a timeline of the species, the life forms of the species through time. So we can see now, it's very interesting, how the total cover varies. So we can see that not always the aerial cover of the species are the same. Sometimes we have higher cover or lower cover, but we never have less than 50%. So we can say that at no point we had like this extreme drought that killed most of our vegetation. So the cover is mostly maintained, and usually dominated by these perennials. But in some years, or in some cases where we have extreme events, we can have a very big decrease in the perennials, and then an increase of the importance of the annuals and the short-lived species. Here is just a figure comparing to those years are not so extreme, but it was the pictures we had. It's to compare the 2012 to 2018, where we can see that there is a decrease. All of these tussock, probably this Festuca valesiaca. And here we basically don't see it, or we see it in very smaller tussocks. Okay, so some of the results I was already discussing before, but now just to highlight what else can I say? That there was in the study basically no general directional pattern of species shift in a clear direction over the last 90 years. So as I told you before, that can have some different outward bias, but it can also mention that the dry grasslands have been stable in this long term. It shifted from one year to another, but basically it's not becoming some different type of vegetation. And over the past 25 years, there was the communities were varying in a synchronized way, but also it wasn't directional. We could see that the synchronization was due to these responses to weather. And there was an exception of this non-directional that was probably these more recent years, 2015 to 18. But except for that was basically no directional changes. And as I told you before, there are similar trends also found by others in a very similar type of environment. And here, just to remember what I said before, that in the long term, the changes were not very clear. And also in these small and fine vegetation surveys, we also just have this small trend in the final years, but in general, they are mostly just very uncommon for. And here, just to highlight again the importance of these non-directional changes for the vegetation. And these non-directional changes, all the temporal changes, depended a lot on these weather conditions. So about the contrasting responses of these species groups, I already told you a bit, but let's recover. The polycarpic perennials are increasing after wet and relatively cold summers. These small, competitive, weak annuals, they increase after moderately warm winters and wet autumns, winters and springs. Here. This share of the species, we can say that they are more ruderal species. They can be of different life forms. You see all the tree forms are mixed in this corner. And they are increasing after these dry and hot summers. And one important thing I want to highlight here is because the winter and the summer weather is not correlated, like it doesn't depend on each other. We can see by the axis that we have, for example, spring here, summer here, that they are basically random. Like if I have like a hot summer now or a wet winter, it doesn't mean that I will have a wet summer or a wet spring. So this, what I'm trying to say is that this increasing species cover can be combined in different ways in different years. So another thing to highlight is the delayed response to weather and shifts in dominance among species group. So as I told you before, we, differently from other researchers, we decided to also take this thin visualization of the weather conditions. We didn't took only the year average or just the growing season average. We decided to go for separated seasons, and not only the year before, but the 2 years preceding the surveys. And we realized, we could see that there was a lag in this response. And this lag is probably due to perennial species because, of course, they are surviving more than 1 year. So what happened today can have a reflect in even 2 years later. Even could be more, but we didn't check for that. And this lag in response can have some cascade effect. Something, as I was trying to say before, about this combination of years and responses of species. Here, just to highlight also specific years where we have rather extreme events. Which was 2003. And we can see here that there was this drought and that the perennial species, they responded here, but they didn't grow all the way in the first year. They took 2 years to fully recover. And this lag in this response was followed by the other types of species that are increasingly a lot following this decrease of the perennials. Here, this trend is even bigger because after this drought and the decrease of perennials, we have a more wet spring, and then we see. And then we see that the short-lived annuals are also increasing, but they are even having higher cover than the perennials. And here also to show the year of 2015. So we could check that in our environment we have this long-term community stability, and it's probably given by these short-term community dynamics. I already told you before, the stabilizing effects of climatic fluctuations can maintain diversity in several terrestrial ecosystems. And they are mostly explained by two mechanisms, which are relative non-linearity of competition and the storage effect. The non-linearity is given by the different shape in the species response to a resource, while the storage effect is to a condition. So it's a very similar idea, but in the relative non-linearity, the fluctuating weather will allow the species to coexist because they will not outcompete each others. And in the storage effect, this fluctuating environment will not allow the species to reach their optimum. Here is a very interesting paper where Adler and others describe very well this storage effect. Of course, they tested with simplified community, or at least they just compare this dominant species in his community and how these dominant are varying through time, but they never manage to outcompete in the sense that one gets extinct. So when the weather is better for this one, for example, the others remain in the community as secondary species or as non-dominant species. And this is why it's called storage, because there is like a storage of this species that can boom when the weather conditions are better. And this is key for maintaining diversity in many ecosystems, and we believe for our ecosystem as well. So some more highlights before we finish. That the stability in our environment are given by means of the interaction of four aspects of the system. The first one is this unpredictable year-to-year fluctuations in weather, the different types of response to weather by the species with different strategies, the interaction among groups of species, the dependence of current species composition on the weather patterns, not only by this year, but from the previous and the last 2 years. And the climatic conditions through the year are important and cannot be simplified just by one or two parameters, such as this mean temperature or annual precipitation, as it's usually done. So we could check that we can have much more detailed results if we separate into seasons. And for future predictions or for future studies, we could check that although there is this increase in the average temperature in the past 28 years, there is no clear directional changes in the species community. And because this complexity and the lack of directional changes, it is very difficult to predict the effect of climate change in the study area. But one thing that we saw is that the summer droughts can increase the abundance of ruderal species. So if there is an increase of droughts, we can have a ruralization of these dry grasslands, and the decrease of the perennial ones, which are usually the most desirable ones or the ones more important for conservation. And it's very hard to see if the stabilizing effects, like the storage effect, will continue to ensure the continuity of this community, because even being a dry grassland, we can see that droughts really affect the community. So it's very hard to say whether it will be stable with extreme weather events or not. So here is, I hope you liked, here is the paper. It's already available for download. It's open source. And it will be published in a special issue of JVS about permanent plots. So the issue is not yet published, but the paper is already available. And something else, as I promised to you, is that we will also publish, when the final version of the paper is published, I believe, in the blog of Vegetation Science, we will publish the history behind the paper about the man who sampled a grassland for 25 years, and his data got eventually published. And this is what he thinks about it. Okay, and as I told you before, now we are working in our second aim of the project, that is this fine time scale dynamics and the phenology, in which we are doing surveys once a month for a year. Actually, the year already finished, but we are still going at least once more. And in these surveys, we record all species cover, height, and the phenological state. And the sampling design is basically, we used the same plots from the previous work, and we are sampling in a smaller quadrat of 1/2 by 1/2 meter for all of the plots we are sampling. And here's just some nice pictures of how the community is varying through the year and how different it is. Here I took specific plots where it's very interesting that you can see species. Even it was funny because some of the rosettes, we didn't know what they were in the beginning, and then we could track their development until they're flowered, and then fruiting, and then it's dead. And you can see still dead here and here. Here is another plot from, it's in another part of the sampling area. And now I ask you a question to guess the richest and the poorest months, because I think you'll be amazed with the response. If you want to turn on your mics and say, what do you think? No? Nobody? Okay. So this is the average richness per month that we have. And March was the richest month. And it's pretty new to me, at least, that we were expecting it to be richer like a bit later in the season. And it's also funny because I would expect the winter to also be very poor, but the poorer months are here. And well, not only the plants have to deal with this stochastic weather, so it was a bit challenging, I believe, not only for me to deal with very cold samplings in the winter, also some very wet samplings, but we also had warm days and we have a lot of fun doing the surveys, a lot of help from many different people. And of course, the climb gets easier every time that I go there once a month. So in the beginning, I couldn't do it in one time. I had to stop several times to breathe. Probably this picture is from the first time that I could do it at one walk. And that's it. Thank you for watching. I hope you are all still there.
Yeah, we are still there listening. Yeah, thank you so much for your nice presentation. I think there will be plenty of questions or comments, and I guess that people just will turn on the microphone, or there is a possibility to add some mark to the chat, and also you can see it. Yeah.
Okay. Now I can see. I'm sorry that you cannot see me, guys, but I just have a problem with my camera 10 minutes before this presentation. So, okay, you can ask.
Is there some question or comment?
No one? Who else is there?
Okay, can you hear me?
Yes.
Perfect. So I really like this box plot showing this dynamics of species richness.
Who's talking?
Jakub.
Okay, your voice is different. Of the richness. Okay, I will put in the presentation.
Yeah. Okay, well.
This one.
Yeah. This is really nice. And did you actually try to figure out how what was the total richness across the season, and which proportion of the richness is captured by each sampling?
Not yet. We just did this.
I know,
morning.
But still, it's cool. Yeah.
But it is a good point. I was thinking about that too. That actually, the richness in March, November, and December is quite bigger due to winter annuals, which dies off after May. But when we look at it, some species are captured in the whole data set for the most of the year, while they disappear in individual plots. Yes, so it would be maybe a little bit more interesting not more interesting, but also quite interesting to look at rarefaction curves for all of the amounts separately.
Yeah.
But we don't have the linked species identity yet. It was just number of observations.
Yeah. Right.
Can you hear me?
Yes.
So so this is Milan speaking.
Yes.
When I look at it, I wonder whether you tried to do an ordination of these data, like whether the difference in species composition between individual plots that are very close to each other on the same slope.
So you are talking about
is larger than the variability in space over the year, variability in time over the year.
Okay, let's see if I understood your question. You are questioning why this Why is not present? Sorry. So you are questioning about, I don't know which ordination?
No, I asked about the data from the second project.
Okay, about the data
monthly
from the
sampling.
Project.
So you showed here this graph for species richness. So
Okay,
I asked whether you also have ordination of these ponds.
Well, so far we just have this. As I told before, we just do it quickly this morning to show something because we were still correcting the data and we had to set, as I told, some of the species we had to go back to see which species it was because it was juvenile and so on. So we took a lot of time to get our definite database. So this morning we basically finished and we could do just this for now. But yes, we can should explore it in several ways now. I don't know. I'm quite wondering what's going to happen.
Yeah, I think that we will need to make a sort of a brainstorming workshop for probably several days to work out what to do with these data.
Then I'm absolutely impressed by this graph because it's so unexpected to me. It's logical when I see it now, but I wouldn't tell it before.
Indeed, it's very surprising.
Well, it is, but well, it shows what's occurring there, because really in summer there is just few species, and they then start to appear as seedlings in October. Yeah, so it nicely shows what's there.
It's actually not correlated with the species covers.
Yeah.
Because the peak of the species cover is between May and June.
Yeah.
And I think the lowest cover values are between November, December, and February. And still in March, the species covers are quite low.
Felícia, there is a question by Padu in the chat.
Oh, let me open the chat.
What is the size of the plots in the figure?
Which one of the figures? Where is the chat? Padu? Which?
Felícia, the ones you were just talking about right now.
Okay, it's 50 by 50 centimeters.
So they are relatively small.
Yes.
Yeah. So is it possible that just at the beginning of the spring, by the end of March, you start to see different species trying to thrive in these small areas, and then just some of them become relatively abundant?
Yeah, we see some of them already flowering like very early in the season. So we have these very short-term lived species. We also have species that start, how can I say, sprouting, or the seeds. You see that we always see seedlings, but not always these seedlings become adults. So we could track this a lot, that there is during the season there is this high amount of seed sprouting, but in the end not all of them will become adults. So I think it's a combination of both. These early season species with these seedlings that are there competing or just trying their chances. Do you agree, Christoph and Jakub?
Yeah.
Yes, of course. It's just fascinating how it's twice as much. The species richness in March and June and July, when the most of the people go sampling actually these grasslands.
Yeah. It will be interesting to plot this box plot actually with like, for instance, seedlings excluded or something like that. Could be interesting. Yeah, to look at different life stages and so on. So there is like a lot of further analysis can be done. Yeah.
May I have a question? You said that you also measure the height of the species. What you plan to do with this measure?
Well, we measure the height to have an idea of this three-dimensional area or volume of the species, and I believe height is also important for competition. Or I can think also about the seeds or the flowers. The height of the flowers are important. But I think that primarily it was more because of the volume to have this idea of not only aerial cover in the horizontal direction, but also in the height.
Thank you.
So I'm Corrado.
Yes.
Something. Usually this kind of people are doing this kind of project in alpine vegetation because there are not so much effect of human. And I would ask you if you consider, for example, the land use, because I don't know. You told before that summer drought increase the number of ruderal species, but usually ruderal species the seeds are coming outside and usually is coming with the animal grazing in the area. So my question is, there is some change in land use during this year, and if yes, you know how to see which is the effect of this land use.
Okay, so the question is about land use and where these ruderals are coming from. The land use is the same for all the plots because they are all in the same area with the same management, which is based on yes.
Maybe can I answer this question?
Yes.
Because this area is unmanaged actually.
Yeah, that's what I was going to say, that the managed is the same that is unmanaged, and some of these wild goats sometimes can go there. And about the ruderals, I'm sorry if I wasn't clear before, but what I was trying to explain is that all the species are there. They're just varying in their cover. So even those ruderals, they are from there. But in these years that are more favorable to perennials, they decrease their dominance. So they only increase when the weather is better for them.
Okay.
So, and we say ruderals because it's like the species are characteristically ruderals, not because they are coming from outside or if we are having some extreme disturbance in the area. Because no, the area is not disturbed. There is no management. There is the natural area.
Okay, thank you.
They are even not ruderals actually. They are more like species with ruderal tendency, but they are, well, basically dry grassland species. Most commonly archaeophytes of some sub-Mediterranean origin, but, well, natively present in those grasslands.
Any more questions?
I have one more question. Well,
yes.
I haven't been there, so I don't know how the setting looks like. But are the plots like one next to the other and relatively close, all at the same distance, or is there some kind of heterogeneity in the way they are set?
Okay, let me just find here. Okay, so we use the setting of IRCAS plots, which are, I don't know how distant you think they are, Christoph, from the 0 to the final one, maybe
around
500 meters.
From the first one to the seventh one, maybe like 200 meters or something.
200 meters. So they are basically in a line following the same altitude around the crest. And it varies. Some of them are like 50 meters away, some are more closer or far away. The distance between the big quadrats are not the same, but for us we use this quadrant as a block. So for us, these inside the same block, they are very close, of course.
So with your answer, do you think there could be an effect also on the distance between plots? Like this is something you didn't consider, right?
We didn't check if the distance between plots had importance. So I cannot say if these ones are close in the field or not. But that's a very good idea. We could check that. That's your question, right?
Yes, it is. It is.
Yes, we didn't test for that. Sorry.
No, it's okay.
Can I have a question?
Yes. Of course.
I very much liked your presentation, and also the paper is great. And I like also the promising results. One more question. Why in the box plot you don't have January? It's the only month I was there.
Okay. Let me explain why we don't have January. You mean January here, right?
Yes, yeah.
Because we were doing all samples in the end of the month, and of course December we didn't went in the New Year's Eve. So we went in the beginning of January. So what you read December here was actually performed in January.
Okay, yeah. Thanks for explanation. I was wondering if you still don't have the data finished or something, or why.
It's finished, but we had some delay also in November. I think it was done in the beginning of December, so it was a bit of delay.
So maybe you can actually put there December, January, like merged month.
Yeah.
Not to confuse people or.
Okay.
But otherwise, well done. Yeah. Thanks.
Okay.
Okay. So, any other questions?
Questions from Brazil? No?
So if not, once again, thank you very much, Felícia. It was a very impressive study and nice paper. Congratulations.
Okay. Thank you very much all. Stay safe, stay well.